Hello, I'm Dr. Mazurki, and I'm here with Professor Tibbs, and we're going to talk about blood typing today. Blood typing is a very useful tool for determining what type of blood a person has for the purposes of transfusions, for the purposes of establishing familial relationships, and it's also utilized in criminal prosecution. But a lot of times people don't really understand what blood typing is and what blood types mean. So we're going to talk about that today. Remember that every single cell in your body, including red blood cells, are decorated with hundreds, if not thousands, of different proteins. And these proteins carry out a wide range of function. The proteins can be involved in carrying out enzymatic reactions, in adhering to other molecules, and in some cases they're involved with the immune system in establishing self or non-self. Now, any protein that is found on the surface of the cell that helps the immune system recognize a self cell, meaning a cell that belongs, is referred to as an antigen. The immune system utilizes antibodies that are secreted by lymphocytes in response to any potential foreign cells. So, in blood typing, we have three main types that I'm going to talk about first, which are the ABO blood types. Now, there's another type of blood, or another typing of blood, that we refer to as either the D antigen or the Rh factor. I'm going to talk about that in a little bit because it introduces an additional level of complexity into the story. So, blood type A is decorated on the surface of the red blood cells with the A antigen. Blood type B is decorated on the surface of the cells with the B antigen. Now, in standard genetics, you learn that things were either dominant or recessive. And so if you had one dominant and one recessive allele, then the dominant trait would, for lack of a better term, dominate. Blood typing is a little bit different, and it is an example of the genetic feature known as codominance. If you have both the A antigen and the B antigen, rather than one dominating over the other, you express them both, and you have blood type AB. Conversely, you can have neither antigen, in which case you are considered to be blood type O. Now, when we test for blood type, we are looking at the presence of an antibody in the plasma and the antigen on the surface of red blood cells. If you are blood type A, in your blood, you will have the antibody to blood type B cells, to that antigen protein that might be on the surface of foreign blood cells. If you are blood type B, you are expressing the B antigen, and your blood contains the antibodies to A. Now, if you're blood type O, you express neither antigen, and you have both the antibodies to A and the antibodies to B. And finally, if you are blood type AB, as you might expect, you express both the A and the B antigen, and therefore you have neither antibody. Okay? This has consequences in terms of performing transfusions, giving and receiving blood. People who are blood type O are considered the universal donor because since they have no antigens on the surface of their blood, now again, this is only considering the ABO blood type. We will introduce the Rh factor, the D antigen, in a few minutes. Blood type O, in terms of the ABO system, is considered the universal donor because it expresses neither antigen on its surface, so it's not going to react to the A antibody or the B antibody in the recipient's blood. So somebody who's blood type A can obviously receive blood type A, but they can also receive blood type O. Somebody who is blood type B, again, can obviously receive blood type B, but they can also receive blood type O, as can somebody who is AB. Now, AB is considered the universal recipient, because since they have neither anti-A nor anti-B antigens, they can receive, obviously, blood type AB, but also blood type A, blood type B, or blood type O. Now, in a clinical setting, of course, we always want to match the blood type precisely to the patient. But there are circumstances where certain blood types are running low, and a decision may be made to give somebody who is an AB blood type either an A or a B or an O blood transfusion if there isn't enough blood type AB around and the person really needs the transfusion. So again, to recap, your blood type is determined by the protein or the antigen expressed on the surface of your red blood cells. And typically, if you are not a codominant individual, you will carry the antibodies to the opposite type of antigen. So again, blood type A has the antibodies to B, blood type B has the antibodies to A, blood type O, which has neither antigen, has the antibodies to A and the antibodies to B, and finally AB has neither antibody. So when we do blood typing, we're going to add the antibodies. We've separated them out. You can buy these things clinically now. This is what we're going to do in lab. You'll add each individual antibody and you're going to see what happens. But first, let's talk about this from a clinical standpoint. As we've mentioned, type A has the anti-B antibodies, type B has the anti-A, that should say anti-A, I apologize, anti-A antibodies. Type AB has no antibodies, so again, they're the universal recipient. And type O has anti-A and anti-B antibodies, so they are the universal donor. This means that if you accidentally give a patient the wrong blood type, so you give somebody who is type A blood, type B blood, they are going to get a transfusion reaction. In this case, what happens is all of the anti-B antibodies, the anti-B antibodies that the type A person has are going to bind to those type B erythrocytes. Remember, an erythrocyte is a red blood cell, and it's going to clump them together. And this reaction is known as an agglutination reaction. And this is very, very serious if it happens in a patient. Addressed very quickly, the situation can be reversed through appropriate medical intervention, but it is a very serious error. And this is why hospitals have gone to great lengths to ensure that the blood type that you are giving a patient is the appropriate type for them. Now, in cartoon form, what's happening is we have antibodies in the plasma. This would be the anti-B antibody. This would be the anti-A antibody. So if a type A person's blood is given to somebody who's type B, these anti-A antibodies are going to start to link all of these red blood cells together, and the same would happen if a type B blood was given to a type A individual, and it will, for lack of a better term, clump up in the blood vessels. This is a systemic reaction. It is serious and will be fatal if not quickly dealt with. In terms of clinical blood typing, what we're going to do in lab, we're going to put a drop of your blood into one of three trays. Okay, so one drop will go into what we call the A tray, one drop will go into the B section of the tray, and one drop will go into the D or RH section. Okay, again, we're going to talk about RH in just a moment. if you're blood type A, when we put the antibody to B in there, you're not going to see any reaction because you don't express the B antigen. However, when we add the antibody to A to your drop of blood, you're going to see a clumping reaction, agglutination. Okay? The opposite happens with blood type B. You get agglutination in the presence of the B antibody and no reaction in the presence of the A antibody. And finally, if you're AB, as you would expect, you're going to get a reaction in both. So blood typing can be done quickly, it can be done safely, it can be done with a minimal amount of blood. We just do a finger prick and get 3 drops out of it. And it gives very clear and very quick results. Now, where does your blood type come from? Most of you probably realize that it comes from your parents, but beyond that, you might not have the background to understand the genetics. So I want to take a few minutes and talk about how we figure out what the possible blood types are for an individual given the parents' blood types. And we're going to use a tool called a Punnett square. Now, before we go forward, I want to talk about what a Punnett square is and the different terms that you're going to be hearing and using as we take advantage of this. So a Punnett square is just what the name suggests. It's a square. In its simplest form, it's divided into 4 subsquares. One of the parents' genetic information goes on the vertical side, and the other parent's genetic information goes on the horizontal side. Now, it doesn't really matter which parent you put where. By convention, we have historically put the mother's information across the top and the father's information along the side. Okay? Now, we need to talk about a couple of different terms now. Okay? The first is the term allele. And this is the term that is most important to understanding what's going on with blood typing. Genetics means that your genes, your DNA, are involved. But what a lot of people don't realize is you actually get 2 copies of every gene. And these individual copies are referred to as alleles. Now, you have 2 copies of every gene because you actually have 2 copies of every chromosome. Remember, we have 23 pair or 46 chromosomes. Now, I'm not going to get into sex chromosomes and the differences that exist there. So for the purposes of our discussion for right now, we're only going to focus on the 22 autosomal, meaning non-sex chromosomes, that are inherited and carried by every human. Okay? Now, these alleles come in one of 2 different versions, possibly, for each individual. Now, it's possible that for whatever trait we're talking about, you are homozygous for that trait. This means that your 2 alleles are identical. You have 2 capital T's or 2 lowercase t's. It's also possible that you are heterozygous, where you have a capital and a lowercase, in this example, T's or G's. Now, what does the case mean? Okay? The case refers to either dominant or recessive. The capital letter indicates a dominant trait. A dominant trait will cover up or dominate the presence of the recessive trait. So in other words, if you are a hybrid, a heterozygote, so in this example, capital T, lowercase t, or capital G, lowercase g, even though the lowercase t is present, the capital T dominates it, and this version of the trait is the only one that's expressed. In the other example, the capital G dominates or covers up the lowercase g, and this version of the trait is the only one expressed. So the only way that you can express a recessive trait is if you are homozygous for that recessive trait. This leads us into another two terms, phenotype and genotype. Phenotype is the physical appearance of the trait for the organism. Okay? So, for example, what it looks like in terms of Mendel's pea plants, and he was the one who first worked this out. You could be tall, you could be short, green, wrinkled, but it doesn't necessarily tell you about the genetics of that trait. Take something very simple like eye color. Let's make it even simpler and limit eye colors to blue or green, or blue or brown, I apologize. Brown is the dominant trait. Blue is the recessive trait. If you see somebody with brown eyes, that's their phenotype, brown eyes. You don't necessarily know what their genotype is because they could be heterozygous for the brown eyes, meaning they carry the brown dominant allele and the blue recessive allele, or they could be homozygous where they only carry the dominant brown allele. In this example of brown eyes, the phenotype does not give any useful information about genotype because there are several different combinations of alleles that that person could have. However, if the individual has blue eyes, you can learn something about their genotype because in order to express the recessive trait, you have to be homozygous recessive for that trait, meaning you have to carry both recessive alleles. If this information is still kind of confusing to you, by all means you can contact me or Professor Moritz, but you can also review this information by looking it up in your textbook. So if you haven't had biology or any sort of genetics explanations in a long period of time, you should go ahead and review this material or learn this material if it's for the first time by reading it in your text first, but then by all means feel free to contact either myself, Dr. Mazurki, or Dr. Moritz. And finally, we talk about ratios, which is when we are looking at genotype and phenotype, what ratio of these different versions exist. Now, all of this is going to sound a little bit clearer when we talk through some examples. Okay, so we're going to do a one-trait or monohybrid Punnett square. In this case, the mother is going to be type B and the father is type O. So, as we said before, the parents' alleles go on the outside of the square. Okay? And this will be, the mother is homozygous for type B, and the father is homozygous for type O, which, being that it's recessive, it would be. Okay? So we put the mother's alleles across the top, the father's alleles down the side, and then we just start dropping the letters on the top into each square. Now, in this example, it's pretty easy. They're all going to be capital B's. Then we move the letters on the side into each square. And again, in this example, it's pretty straightforward because they're all lowercase i's. Now, the order that you arrange them doesn't really matter, but by convention, we tend to put the capital letter or uppercase letter first. So what does this show us? Well, B is the dominant allele for the B antigen, and I is the recessive allele for no antigen, or blood type O. So we can start to make predictions about outcomes. And this is the important thing to understand. These are predictions. If this mother and this father conceive a child, this is simply outlining the blood type that that child may or will have. In this example, the phenotype is 100% blood type B because you have a 4:0 ratio B to O. Okay? The genotype is also blood type B because you have all of the B and then O or no antibody, antigen rather, alleles. So this is a very straightforward outcome. But keep in mind, if you deal with somebody who is blood type B, you don't necessarily know if they're heterozygous or homozygous in this instance. Now, if we go forward and we look at another Punnett square, where now we have a mother who's homozygous blood type A, a father who's homozygous blood type B, and I apologize, the letters are kind of dropping down a little funny. We again end up with 100% AB. Now, this is an exception to the dominant recessive rule, okay? Because in this case, rather than A dominating B or B dominating A, they're both being expressed. And this is an example of codominance, where both things are expressed. So if you are blood type A, you're only expressing the A antigen. If you're blood type B, you're only expressing the B antigen. But if you are blood type AB, you are expressing both of them equally. So that's codominance. Now, let's talk about the Rh factor. Rh stands for rhesus. We also use the letter D to represent it. So if you're Rh positive, you have the antigen, D or dominant. If you're Rh negative, you do not have the antigen. So you have the lowercase d and it's recessive. Now, what's interesting is an Rh negative individual will also not have the anti-D antigens unless they're exposed to Rh positive blood either through a transfusion or through pregnancy. And we'll talk more about that in just a moment. But let's look at this additional level of complexity for blood typing. So this is completely independent of the ABO system. This is simply saying if you have or don't have the D antigen. So looking at our Punnett square, we've got a positive mother and a positive father, but they are both heterozygotes. So when we start dropping our letters down, we get a different type of ratio. Okay? Remember, D is the dominant allele for the Rh antigen, and lowercase d is the recessive allele for the Rh antigen. So in this case, we end up with 75% of the offspring potentially being Rh positive and a 25% chance of any given offspring being Rh negative. Now remember, this is statistics, so these are probabilities. These two parents could have 8 children, and those 8 children could possibly be all homozygous positive or all homozygous negative. Now, that's not likely, but it is possible. For any given pregnancy, these are the likely phenotypic and genotypic outcomes, where you would have one homozygous dominant or Rh positive child, two chances of a heterozygous, also Rh positive child, and one chance of a homozygous Rh negative child. So our ratio is 1 to 2 to 1, 25% homozygous dominant, 50% heterozygous, 25% homozygous negative. Again, it's all just odds or probabilities. It's not until the sperm actually merges with the ova that one of these is defined. So this is simply saying the possibilities. Now, as we said before, in general, Rh negative people do not have the Rh antigen, antibody, except in the case of an Rh negative mother and, in this example, a heterozygous Rh positive father. Okay, we put in our individual letters and we find that this mother and this father have a 50% chance of giving birth to an Rh negative child and a 50% chance of giving birth to an Rh positive child each time that the mother becomes pregnant from this father. So our phenotype is 50% Rh positive, 50% Rh negative, and our genotype is going to be 2 of the heterozygous positive and 2 of the homozygous recessive. So, what does this mean for this couple in terms of having multiple children? A lot of this traces back to events that happen in childbirth. Okay? Childbirth can be a very messy event. A lot of things can go on, and there's a possibility of the mixing of the maternal and the fetal blood. Now, up to the point of childbirth, this first Rh positive child in the Rh negative mother is going to be fine, because unless she had a bad transfusion or something, this Rh negative mother doesn't have the antibodies to the Rh positive antigen. So the fetus and embryo are able to develop normally. Up until and after the first trimester, the blood supplies are separate. So there's no mixing of the maternal and the fetal blood, because they're 2 separate supplies that separateness is maintained by the placenta. It's not until the point of birth that there's a potential for mixing of the blood. If that happens and the mother is exposed to Rh positive blood, her body will generate the antibodies to the Rh antigen, that D antigen. All subsequent pregnancies would therefore carry a risk, because if she became pregnant again with an Rh positive embryo, this embryo would immediately be attacked by her body, because she would now have the antibodies circulating, and it would most likely be destroyed prior to the end of the first trimester, where the blood supplies become separate again. In order to deal with this, we now treat Rh negative mothers with RhoGAM, and this inactivates the antigens. Okay, this is done preventatively in an effort to ensure that there is no problem with any subsequent pregnancy. This same blood typing discussion applies to the exercise in your lab manual, Who's the Baby Daddy? So now that you understand how blood typing works and how the genetics work, you can go forward and do the blood typing exercise, Who's the Baby Daddy? We will be doing, in my lab, blood typing and hematocrit measurements and hemoglobin measurements in a future lab that I've discussed in the announcement on Blackboard. Thank you very much for your attention. This slideshow will also be up in the Dr. Mazurki lab section of Blackboard with the corrections that I missed in putting this together, and again, I apologize for that. And I or Professor Moritz can answer any of your questions. I look forward to seeing my lab again next Friday when hopefully we have power. Have a great day.